Right, so welcome to genetics portion of ACP Bio. So I'm going to try to get through this part pretty quickly here because I do have several examples for you guys to work through. Hopefully, there may only be like a couple new things. We're going to be doing some monohybrid crosses, so just those basic Punnett squares, dihybrids, which when we have looking at two traits, and then we're going to look at product rule. So that is probably the only thing that you probably haven't covered yet. So we're going to get just into a little bit of background in what is heredity, what is genetics, and talk about Mendel a little bit, right? You probably heard of Mr. Mendel before. Okay, so heredity, when we're looking at heredity, this is studying how genetic information gets passed from parent to offspring. So we know on a molecular level, that's because we've got meiosis happening to form those gametes, and those gametes combine during fertilization. All right, and then genetics specifically is the science of heredity. So this is really looking at, what does it mean physically for individuals because those chromosomes are combining. So how does that impact our actual physical features? So some words that hopefully you guys are familiar with. Hopefully, like I said, what's nice about today, even though I'm not with you, even though we're just doing a flip video on this, these are things that you should have already heard of, you should already know. So hopefully all of this is pretty much a review. So phenotype, when you think of phenotype, you should think of physical, right? So phenotype, this is the physical appearance of an organism. What do organisms look like? So this is the expression, the physical expression of a trait. So for instance, eye color, right? What do we see? We could have blue eyes, we could have brown eyes. I'm going to simplify a lot of these things. We're going to look at one organism in particular because humans, I'm going to say this probably a lot, humans are rather complicated. Most of our traits aren't this straightforward, but we'll try to hit on some of the ones that might be. All right, genotype, the genetic makeup of an organism. So when I'm talking about the genetic information, I'm talking about the alleles. Okay, so the alleles are things that represent the different options for the traits. So we have, we typically represent them by dominant alleles and recessive alleles. So we've got three different possible genotypes here. And so we know we have homozygous dominant because both of those are the same, heterozygous, and we don't say heterozygous dominant, we just say heterozygous, or homozygous recessive. All right, and then we get these cute little doggies here. So brown eyes is dominant over blue eyes. All right, so talking about Mendel. All right, Mendel is considered the father of genetics. He was an Austrian monk. I like to say that he hung out with bees because he couldn't talk to anybody else. I don't know that. I just assume that because he's a monk, right? He took a vow of silence. I'm just being silly. So he actually experimented with over 28,000 peas. So he's actually a great scientist because he has tons of data. And he had 28,000 peas over 8 years. So what's interesting is he actually isn't really trained in science. He's actually more of a math person. He's more into statistics. So luckily for us, he did all this work and he figured out the statistics of, what he was seeing. So he applied these quantitative methods to studying inherited. So we had these really well-planned experiments that he looked at and recorded detailed information on what he got and then kind of worked backwards to figure out what was happening. So one of the things that we need to know about that Mendel discovered from his experiment was this idea that some traits are dominant and some traits are recessive. So remember we talked about these are the alternate options for the genes that represent the phenotypes. So dominant, I'm sure you've heard of this before. This is going to be represented by a capital letter. And this is going to be the trait that shows up whenever it's present. So it masks or hides the recessive. Recessive, this is going to be the masked trait. So in order for you to get a recessive phenotype showing up, you have to have 2 recessive alleles. Okay, so when we think about these alleles, right, remember, where are you going to get those alleles from? You end up with 2 alleles. Where are they coming from? your parents, right? One came from mom, one came from dad. These alleles are traveling on those chromosomes in the gametes, right? And they combine to make the offspring. All right, when we talk about hybrid, this is another name for heterozygous. So this is something I might say on your problems that you're doing. You need to know when I'm talking about a hybrid, I'm also talking about a heterozygous individual. So when this is a little bit different because we're talking about a hybrid between two different species. They also use this in this context. So we've got a zebra, we got a donkey, and then we got a zedonk, right? Zedonk. This is zedonkulus. I hope you laughed at that. I'm sure you didn't, but I did. All right, so let's talk more about Mendel and kind of how he came to his conclusions. So you need to know a little bit about Mendel and his peas. So what Mendel first started doing is he first started with true breeding or pure breeding strains. So that's another term you need to know. When I talk about purebreds, that means individuals that are homozygous. So he took those purebreds. These are individuals that over and over and over from one generation to the next, they're always going to produce the same phenotype. So we have their grandparents produced purple pea plants. Great-grandparents produced purple pea plants. Keep going back, all right? So we had these generations. We've got this purple pea plant that always produces more purple pea plants. And then we've got a white pea plant that always produces white. So we have these two homozygous or purebred individuals. And what he's going to do is he's going to decide who the parents are. So he is going to use something called cross-pollination in the parental generation. So we also call that the pea generation. These are the parents, okay? Normally what happens is a pea plant can self-pollinate. So it's just the pollen, aka sperm, it's just going to fall on itself. And so you have the sperm and the egg combining within the same pea plant. So technically, I know this is kind of crazy, but these plants are doing sexual reproduction because we have two different gametes combining. So they're not clones because we have two separate gametes combining. So they're technically doing sexual reproduction here. But normally what happens is it's self-fertilization. So what he's going to do, oh, sorry, I thought I'd get into this a little bit more. He is going to cut off the sperm-producing parts, cut out the pollen-producing parts of one plant, and he's going to take the pollen from the other plant. So he is going to control who the parents are. So this might be the dad. He takes some pollen from here and puts it on this plant right here. And so he's going to say, here's the dad, here's the mom. All right, and what he saw over and over again was when he crossed purple plants and white plants, they always showed up purple. So the white disappeared. This is called the F1 offspring. So the F1 offspring were always purple. So 4-0, purple to white. Then he's going to let these self-pollinate. So he's going to let this plant right here, this plant is going to, the sperm, the pollen is going to just fall on this plant. And so we're going to see what happens. So we have two of the same plant. They have the same genotype technically. We're going to see what happens in their offspring. And so what he noticed over and over and over again, when we get to the F2 generation, it was a 3-to-1 ratio. This white trait reappeared, right? This white trait reappeared. So this is really what happened. Okay, so this is our monohybrid cross. Mono means one, so we're looking at one trait. So this is the F1 generation. So it was self-pollinating. So here is, they were heterozygous because remember they had one bee coming from the purple parent, one white allele coming from the white parent to get you the F1 generation. So we self-pollinated those, and this is what happened. So this is why the white reappears, okay? Because we had two recessive alleles recombining, and the recessive shows back up. That was Mr. Beaver calling, so you can yell at him later. All right, so there's our monohybrid cross. So make sure when we're setting these up, I'm sure you've done these over and over and over again. So this should be really simple for you guys, but remember each of these boxes represents a gamete. So for parent 1, we separate those. We go from dihybrid to, or not dihybrid, diploid to haploid when we form our gametes. So this is gamete, this is gamete. All right, and same thing here. And we bring it down, we bring it over. And let's see. Okay, so why did Mendel choose peas? One of the things that's nice about peas is that they're easy to observe their traits. Okay, it's easy to see, purple or white, round or wrinkled, tall or short. All right, so that's helpful. Okay, so here are some of the traits that he studied. All right, and you can see we end up having the dominant on the right and the recessive on, or sorry, dominant on the left, recessive on the right. All right, and then what he saw was, this is kind of what his data showed him over time. So you saw pretty much, when you simplified it, over and over and over again, pretty close to a 3-to-1 ratio. There's a little bit of drama there because supposedly were his results a little too perfect? We're not sure, okay? But they've repeated the experiments and they've gotten the same thing. But the thought is that he may have manipulated the data, so there's a little bit of drama for you. All right, another thing that's nice about pea plants is they can cross-fertilize, right? We can choose the parents. They can self-fertilize, okay? So we can use that to our advantage. And also it's quick, right? You can grow a lot of pea plants in a small space. They're easy. They have lots of offspring. We can go from one generation to the next very quickly versus humans, right? If we have a baby, it's going to take a while before the next baby comes. And we don't have a ton of offspring. So we have to actually look at, we're going to talk about pedigrees and how we use pedigrees actually within humans to try to figure out what's happening. All right, so we talked about the law of dominance and recessive. So dominant traits mask the recessive traits. And then we have 2 more things I'm going to try to get in. The principle of segregation. So this idea that alleles for traits, only one allele for a trait goes into a gamete. So like we talked about, the big B and the little b, those are going to separate. That's called the law of segregation or the principle of segregation. Which way those go is random. This is why you flip that coin, right? To decide which allele was going to which spot when you separated your chromosomes into chromatids. This happens during anaphase, right? So that's random. Another thing, you got to get the difference between these, segregation and independent assortment. So independent assortment, this is the idea we're going to separate alleles for different traits. Okay, so the way these chromosomes get arranged can be different. So they're going to be different. This is why, once again, at the beginning of meiosis 1, remember when you lined up your chromosomes, when you lined them up, you flipped a coin to decide, okay, is the mom's going to be on the left or is the dad's going to be on the left? Okay, and we flipped that coin 2 times. We would flip that coin for every pair of chromosomes. It's also random. Okay, so this is observed, the way they separate is going to differ during meiosis 1, and that's going to impact what we end up with. Okay, so principle of independent assortment, alleles that are found on different chromosomes are going to separate from each other independently. So this idea, we may have the gene for green and yellow, right? And so just because you have green peas doesn't mean that you're also going to be tall, all right? These can be separate because they're on separate chromosomes. All right, so here we go. Here's some good pictures here to make sure you understand the difference of what's going on. So segregation. These alleles for a chromosome are going to separate from each other. And then independent assortment, the way they line up, here's possibility 1, here's possibility 2. Okay, that can change our genetic variation. All right, so test cross, I'll let you write this down. Pause, I don't have a ton of time, so pause this, write this down on your worksheet. I'm going to go through a problem in the next few. Dihybrid, write down our goal here. Pause that, write that on your worksheet. All right, foil, we'll go through that. Foil, dihybrid problems, we're going to talk about that. And then product rule, go ahead and pause and write this definition down so you know what that is. All right, and we'll go through that in the videos. And that's it.